Many years after Jenner, till the formulation of germ theory of disease, the field of vaccination did not show any developments. It was Louis Pasteur who, after contributing to the development of germ theory of disease, came to devise the methods to prevent the disease. To prove that he had isolated the causative agent of cholera, he arranged a public demonstration. He inoculated the healthy chickens with his pure cultures, but the chickens did not die. He then discovered that the cultures he had used were several weeks old. He repeated the experiments using 2 sets of chicken. One set included the chickens inoculated in the previous demonstration with the old cultures, and the other which were not previously exposed. Both the sets were inoculated with fresh cultures. In this experiment, the second group died of cholera while the first one survived. This confirmed that if by some means the bacteria or the causative agents were attenuated, here by using the old cultures, they lose their ability to cause the disease but retain the ability to stimulate the immune system to produce antibodies. This gave a plausible explanation for Jenner's methods of providing protection against smallpox. Pasteur was able to successfully isolate Bacillus anthracis, responsible for causing anthrax in cattle. During his studies on anthrax, Pasteur announced the discovery of vaccine against anthrax. The public demonstration of the vaccine was to be held at Pauley-le-Fort on 5th of May. 50 healthy sheep were selected, out of which 25 were vaccinated and the others were kept as controls. The vaccinated sheep received the second dose on 17th of May. And finally, all 50 were injected with the virulent strain of anthrax bacilli on 30th of May. On the 2nd June, all the uninoculated control sheep died of anthrax while the other survived. Thus, the vaccine for anthrax came into existence. The same principle was applied by him to produce the vaccine against rabies. Hydrophobia or rabies is transmitted through bites of dog, cats and other animals. Pasteur was able to successfully immunize a young boy, Joseph Mister, in 1885 against rabies. Joseph was bit by a rabid wolf, and since there was no treatment available, his family was ready to take a chance with Pasteur's vaccine, which was not tried as yet. It worked. Rabies-causing organism was never grown in the laboratory, but the disease was produced in the rabbit by inoculating with the saliva from the rabid dog. The infected brain and the spinal cord were dried for several days. This was crushed and mixed with glycerine. This made the vaccine which was given to Mr.